Prokaryotic metabolism is highly diverse. So by highly diverse, prokaryotic organisms include every type of nutrition that we know of. So they have all of the same types of nutrition capabilities that we have in eukaryotic cells, plus they have some of their own methods as well. So if we look at this table here, this is a table from your textbook. This lists the different types, or just some of the major types of nutrition that we have in prokaryotes. We have what are called autotrophs, and these are the ones that are self-sufficient. So they don't require outside nutrients in order to survive. And then we also have what's called heterotrophs. And heterotrophs require some organic nutrient. So if we look and talk first about the autotrophs and the most common autotroph examples. Photoautotrophs are able to take energy from a light source, and they're able to use that to synthesize organic molecules. A good example of this is any type of photosynthetic bacteria. Cyanobacteria are a big one here. These are able to take light energy, so from the sun, and they're able to use that light energy to take inorganic carbon, which would be carbon dioxide, and convert that into sugar molecules. Chemoautotrophs, chemo means chemical, these are able to use chemical energy. So they're able to take inorganic molecules such as hydrogen sulfide or just iron, and they're able to use those to fix organic molecules. If we talk about the heterotrophs, what heterotrophs will do is they're going to have to take in some ready-made organic molecules. So by ready-made, these are ones that have been synthesized by autotrophs. So they're something that they might find in their environment. We can have photoheterotrophs. Photoheterotrophs would use light as an energy source. We can have photoheterotrophs. Those would use light as an energy source. And we can also have chemoheterotrophs, and they would use organic compounds as their energy source. So that's just a brief overview of the different types of nutrition that we have in prokaryotic cells. There's certainly more than what we're seeing listed on this slide. These are the most common ones. Another thing about prokaryotic cells is that they are very wide-ranging in their use of oxygen, or in the way that oxygen plays a role in their metabolism. Here's 3 different types of prokaryotic organisms. We have obligate aerobes. Aerobes means oxygen using. So these are ones that must use oxygen. And by oxygen, we're talking about oxygen gas, which is O2. So without oxygen gas, these obligate aerobes, they would not even survive. So they must have oxygen gas for their metabolism. We have obligate anaerobes, and since these are anaerobes, these can't survive at all with oxygen present. So these would need to live in places where there is no oxygen around. So you might find these in swampy areas where you have limited oxygen present, but certainly different environments from what you're going to have humans and other aerobes living in. Obligate anaerobes would have been some of the ones that would have been greatly harmed when we started to have an abundance of oxygen gas in the atmosphere. Facultative anaerobes are organisms that can live either way. So they can use oxygen, they can do without oxygen. So they adjust their metabolism according to the environment that they are living in at the moment. We talk about the nitrogen metabolism in prokaryotic cells. They have a wide range of abilities as far as nitrogen is concerned as well. They're diverse in their ability to utilize different forms of nitrogen. They can perform what we call nitrogen fixing. And with nitrogen fixing, we're taking atmospheric nitrogen, which is N2, and they're able to convert that into organic molecules. We have them able to use things like ammonia or ammonium, but very diverse metabolism with nitrogen. We also have metabolic cooperation between different cells. In general, our prokaryotic cells are going to be unicellular, but oftentimes we find them living in very close-knit colonies. And when they live in a colony, the different cells of the colony can specialize for different aspects. That's what we're seeing in this particular slide. This is a cyanobacteria, and this particular one is able to perform photosynthesis. It's also able to fix nitrogen. There are specialized cells, which are the larger ones that you're seeing here, and these are what we call heterocysts. And what these specialized cells do is they fix nitrogen. So they're able to take in nitrogen gas and convert that into an organic form that the neighboring cells are able to use. What is going to happen in all the rest of these cells is these are going to perform normal photosynthesis. And what photosynthesis does is it fixes carbon or carbon dioxide. So we've got cooperation because we've got some cells that are performing one activity for the benefit of the other cells, and then we have our photosynthetic cells which are performing that for the benefit of the nitrogen fixing cells. We also have examples of what we call biofilms. Biofilms are many big colonies of prokaryotic organisms. They live together in close proximity to each other and they work together. So you can have something synthesized by one group of bacterial cells in the biofilm, and they actually eject that so that it can then be taken up by other members of the colony. If we talk about the classification of prokaryotic organisms, we have to go back to this phylogenetic tree. This phylogenetic tree shows that we have all organisms arising from one initial ancestor, and then we ultimately ended up with 3 domains of life. Well, 2 of these domains, so 2 out of the 3, are going to be prokaryotic organisms. So the domain Archaea, those are prokaryotic organisms. Those happen to be more closely related to eukaryotic cells than they are to the 3rd domain, which is also going to be prokaryotic, and that's the domain bacteria. When we typically talk about prokaryotic cells, we're usually focusing in on the bacteria ones, but we do want to keep in mind that that Archaea domain exists as well. Now, this table from your textbook just shows a comparison of the 3 domains with some of their major characteristics. If you go down this list, you can see that there are a number of categories or areas where the Archaea and the bacteria match each other. And then you can also see a number of categories where the Archaea and the eukaryotes match each other. So take a look at this table. Do be familiar with the characteristics that you see on here as far as which ones you find in bacterial cells and then which ones you would find in archaeal cells. If we talk about Archaea first, Archaea are a lot less familiar to students. And one of the big reasons for this is that Archaea tend to live in very extreme environments. So what we mean by extreme environments is that these are environments where normal organisms do not survive. So these organisms are what we call extremophiles. If we talk about some of the extremophiles, we have extreme halophiles. These live in very salty environments. So the Dead Sea, the Great Salt Lake, those would be places where you find these. We can find Archaea living in environments where you have as high as 30% salt. We also have extreme thermophiles. These thermo is a temperature thing, so these can live in very cold or very hot environments. So these would be some of the prokaryotic cells that you might find in the Arctic regions or in Antarctica. We could also talk about some of the hot springs that you might find out west, say in Yellowstone. So these have very high temperatures, and those are temperatures that would kill most organisms. but these are extremophiles because they're able to not only survive in those environments, but they're able to thrive in those environments. The methanogens is a special category, and these are able to use carbon dioxide to oxidize hydrogen gas. And what they do, and the reason that they have this name, is that they are able to produce methane, which is CH4, as a waste. These are organisms that are going to be obligate anaerobes, meaning that they can't have oxygen gas or can't live in the presence of oxygen gas, so they're actually poisoned by oxygen gas. And then this means that they're not going to be then present in our typical oxygen-rich environments. So these are things that you would find in swampy areas mostly, or marshes. So a lot of diversity as far as where you would find Archaea. We said that a lot of times they're found in extreme environments. This does not by any means mean that that's the only place you have them. They can be found in normal environments that you would find other bacterial cells, but this is what they're typically known as. So they're known for living and surviving in these very extreme places. If we move on to talk about the bacterial domain, there's a lot of diversity with bacterial species and the ones that have been classified. Many more remain to be classified. So we're really just looking at a small sampling of bacteria here. But altogether, bacteria have been broken down into 5 major groups. The first group is what we call proteobacteria. And proteobacteria are going to be gram-negative bacteria. So that's what they're mostly known for, is the fact that they are gram-negative. We have different categories within the proteobacteria. The alpha proteobacteria, these are known for their nitrogen fixing capabilities. When we're talking about the betas, these are very diverse with their metabolism, but they're better known for their ability to oxidize ammonia gas. We can also talk about the gamma ones. These are able to use sulfur. So they are able to oxidize hydrogen sulfide. The delta ones include a lot of kind of attack bacteria, so ones that are able to attack other bacteria. And then the last one is best known for lots of pathogens. Okay, so these are gram-negative bacteria. Gram-negative, if you'll recall, these were some of the ones that are the more toxic bacteria. So better known for being very toxic because they do have those liposaccharides on that outer layer of their cell, and those make it a lot harder to actually target them and treat them when they do infect some organism. Here's just a look at the other major groups that we have with bacteria. The chlamydias, these are ones that can only survive inside a host. So they must live inside a host. There's a number of diseases that are caused by chlamydias. We have spirochetes, which are really named according to their shape. So notice that they are helical or spiral in shape. We have cyanobacteria. These are more known for their photosynthetic ability. These are thought to be the ancestors of chloroplast, once chloroplast did evolve. And then we have the gram-positive bacteria, which are just as diverse as the gram-negative bacteria or the proteobacteria. So the gram-positive, very diverse. And these include even what we call the mycoplasmas. I mentioned the mycoplasmas because those are kind of an odd group. Those actually lack cell walls. So that's something that's very unique for a prokaryotic cell, or unique for bacterial cells. So with all of this diversity of prokaryotic organisms, they have a lot of ecological impacts. So there are many of them present all around us. If we talk about some of their ecological impacts, they are very good, many of them, at chemical recycling. So they play a very important role as decomposers in our environment. So taking dead and dying materials and converting those into products that can be reabsorbed by organisms. We also have them forming a lot of symbiotic relationships. There's a number of these. We can have what's called mutualism. With mutualism, you have both participants benefit. So this would be good for both parties that are involved. We have commensalism, where one participant benefits. Both of them benefit, one of them benefits, but neither one of them is going to be harmed in the process. And then we have parasitism, where one benefits, but even more than that, one benefits and one is harmed. So that would be what we consider a lot of diseases that are caused by prokaryotic cells. We talk about the effect that prokaryotic cells have on humans. We have about 500 to 1,000 species that live just inside our intestines. So we definitely have tons of bacterial cells inside of us every day. This is just talking about our intestines, but you would find them all over the place inside the human body and on the surface of the human body. They can be mutualistic bacteria. We have ones that help us to digest food. They can help to maintain the proper pH in locations. So when we talk about bacteria, these are not all harmful bacteria. So mutualistic, these would be ones that are good. We can also have pathogenic bacteria. So about half of human diseases are caused by some type of bacteria. So that's quite a lot of them. Some of these can be simply things that are going to be uncomfortable but recoverable for a human. Others are going to be quite deadly for a human. So how do prokaryotic cells cause disease? They have a number of different mechanisms that they can use. First off, they can be capable of invading host tissues. So when they invade host tissues, that can cause a problem right away, but it can also cause a problem because they can produce toxins. So they can produce molecules that they secrete that then affect other molecules inside of our cells or inside of our tissues. There's different categories to these toxins. We can have exotoxins. These are ones that are actually secreted, so they exit the prokaryotic cell and then they affect the cell that they are in or next to. And botulism is a good example of this. We can also have what's called endotoxins. And with endotoxins, this is the outer membrane, that liposaccharide portion of the prokaryotic cell that is causing a negative reaction with the molecules there. So this is going to be the gram-negative bacteria that produce the endotoxins, because the gram-negative bacteria, those are the ones that have those liposaccharides on the outside. As far as research and technology goes, prokaryotes play a big role there as well. So these would be advantages to having prokaryotes around, or some of the major benefits that we have to human society from prokaryotes. First of all, they can be used in biomedical research. They can be used, and there's a lot of research that goes into trying to use them to produce drugs. So they can also be used to test different chemicals and see what the effects might be. They can be used as metabolic factories. They could be used to produce something like a particular plastic. They could be used to produce something like insulin, a drug. They can be used for bioremediation. This would be them being used to clean up areas in the environment that might have experienced an oil spill, for example. Some bacteria are very good at breaking down oil. So if you take these bacteria and you spray them on a place where we have had an oil spill, that oil will get cleaned up a lot faster. The last one that I want to point out is their use in molecular biology. So they are very small organisms. They have very fast generation times, and they have a relatively small chromosome. So this makes them a really good organism to use to manipulate DNA, study the effects of different changes in the DNA. And we see E. coli being used for this a lot, but there's other bacteria that are used in molecular biology research.